Fred's nervous now because I'm pulling out my notes that I get to introduce him. It's only like 12 pages. If we could, oh, it's there. I had to do this. Okay, so our keynote speaker is Dr. Fred Gould. I have a few notes of introduction. I'm not assuming everybody here knows who Fred is, so I'm going to take a few minutes to introduce him to you. Fred was born and raised in New York City, and he attended Jamaica High School in Queens, New York. So he is a New Yorker through and through. He earned a bachelor's degree, a BS in biology from Queens College, City University of New York in 1971. During college, he was a pre-med student and a self-proclaimed hippie. He applied to medical schools after college, and he did get in, but decided that was not for him. Instead, he bummed around Europe, he hitchhiked across the United States, he drove cabs in New York City, and worked in a psychiatric hospital. Eventually, he grew weary of the 9-to-5 routine and went to State University of New York at Stony Brook to earn a PhD in ecology and evolutionary biology. His thesis examined rapid host range evolution in a crop pest, and he finished graduate school in 1977. In 1978, he joined NC State as a research associate, and a year later was, I believe, was an assistant professor in the Department of Entomology. He has been at NC State ever since. Fred's research applies evolutionary biology and population genetics to enable sustainable use of insect-resistant crops and genetically engineered agricultural pests. He also does research on strategies for development and use of engineered mosquitoes to decrease human disease. Working in these areas led him to a significant interest in how engineered species are utilized and regulated. In 1993, he was named William Neal Reynolds Professor of Agriculture here at the state. In 2002, he made this official connection to genetics because he was an adjunct professor in the genetics department when we had a genetics department. In 2011, he was named University Distinguished Professor and elected to the National Academy of Sciences and elected as Fellow of the American Association for the Advancement of Science. In 2013, he was an inaugural co-director of the Genetic Engineering and Society Center at NC State. In addition to the multitude of research grants Fred has worked on, he has been the director of 2 multi-million-dollar NSF graduate training grants. The first grant was titled Genetic Engineering and Society, The Case of Transgenic Pests. The second was Agricultural Biotechnology in Our Evolving Food, Energy, and Water System, affectionately referred to as Ag BioFuse. Fred has served on National Research Council committees addressing regulation of genetic technologies in agriculture, and he received the Alexander von Humboldt Award for Most Significant Agricultural Research over a 5-year period, the Sigma Xi George Bugliario, I probably pronounced that incorrectly, sorry George, Prize for Written Communication of Science, and the O. Max Gardner Award in 2012 for the UNC faculty member with the greatest contribution to human welfare. He was elected to the U.S. National Academy of Sciences in 2011 and served on the NRC Board on Agriculture and Natural Resources. He has given invited presentations and keynote speeches at countless universities, conferences, and workshops. His CV I was going through last night, there was just no way I could list everything he has done. NC State has been lucky to have Fred as a faculty member for over 45 years, and while at State, he has taken on numerous leadership roles throughout his career, with perhaps his most recent being as the executive director of the GGA and co-director of the Genome Engineering and Society Center. I have had the privilege of working with Fred for the past 6 or 7 years, first on the Genetics and Genomics Initiative, and most recently, the GGA, the Genetics and Genomics Academy. In that time, I've gotten to know him a bit, and my top 2 takeaways about Fred are, number 1, his joy for life, which you can see it when he smiles, and number 2, his compassion for other people and for the future of the planet. It is with great honor and gratitude that I introduce our keynote speaker today, Dr. Fred Gould.
All right. So for all those graduate students out there, after 45 years around here, I still get nervous giving a talk like this, especially to all these people that I know. So the first thing is thanks, and thanks to Jeff for that nice introduction, and also for taking over the leadership of the GGA. I know how much work it is, and I bless you. So I think that's wonderful. I've seen what's been going on today, the intellectual rigor of this whole day, as well as the humanity of this whole day. And I think this is a wonderful thing to see. And I look forward to watching it from the sidelines. So I also want a shout out to the executive committee that I've worked with for so long, and now Jeff is taking that over. This bunch of people has been just tremendous in meeting almost every week for whatever it is, something 4 or 5 years. That's why we have this. So on to this talk, and I hope that I'll get through it by 5 o'clock and let you guys go. But, so why am I talking about genetics in society, past, present, and future? And I think I'll explain that. Is this microphone and everything work if I move over here a little bit? So if any of you have been watching our website over the past few years, you'll see our tagline, genetics, genomics, and you. And if we focus in on this a little bit, you'll see that I think it's called a mission statement that says the Genetics and Genomics Academy promotes and supports collaborations across all research, training, and outreach activities at NC State University to forge a more interdisciplinary understanding of the genetic and genomic underpinnings of life on Earth to sustain biodiversity and improve societal equity and well-being. That is a major thing. So just to focus here on that, to sustain biodiversity and improve societal equity and well-being. That is our mission. And that is a challenge. That is a real challenge in today's world. I'm going to focus more on not the biodiversity in my talk, but more on the societal equity and well-being. And the reason I'm giving this talk also comes from what I have learned from being involved with the Genetic Engineering and Society Center, which collaborates closely with the GGA. And their tagline is integrating scientific knowledge and diverse public values in shaping the futures of biotechnology. You can see how these two missions come together. And Jeff's already been reaching out and pulling in more of the people from the Genetic Engineering and Society Center to function with the GGA. And this is my perspective here. This talk is going to have two parts to it. First, I'm going to talk about responsible innovation in genetics and how the social sciences and humanities can help us to do a better job of that. And then I want to really get into talking about dealing with genetic determinism and what the role of the GGA is in dealing with this. So in terms of responsible innovation in genetics, what I want to focus on is use 4 case studies that have involved NC State members, present or past. And to start out with, I want to bring up this saying from Neil deGrasse Tyson, because as a researcher, focusing a lot on doing strong genetic research that has application, this appealed to me so much when I first saw it, right? The good thing about science is that it's true whether or not you believe in it. And that's it makes us focus and think really great things, but I've come to having a different perspective on this. And I want to show that I was challenged a lot in getting involved with the Genetic Engineering and Society Center. And there was one paper that we read and had a conference about that was really important to me. And this is a paper published in Nature in 2015 called Science Can't Solve It. And the interesting thing for me was that when I first read this paper, I hated it. I thought, what are you telling me? That's what we've been doing all these years. social scientists can tell us we can't solve it. We know how to solve problems, but it's grown on me. And the good thing about hating something like that, not just dismissing it, but hating it, is that you have to live with it. And one of the reasons for hating it is because it had a point in it that you didn't want to believe, but you had to then take into account. And what they're really saying in this is democratically weighing up the benefits of risks here about gene editing and artificial intelligence is a political endeavor, not an academic one. And to accept that and understand that, I want to give you one example, and that is the COVID vaccine. Think about that. The incredible ingenuity of decades of work to develop this vaccine. As an older citizen, I might not be here if it wasn't for the COVID vaccine, right? It's an incredible thing. But then you think of what happened to COVID and the rollout of the vaccine in different countries and different things. There are huge decisions that had to be made once that vaccine was produced. And hello, you weren't involved in that as scientists. And sometimes as scientists, I look at this picture and think, wait, everybody's thinking that it's the scientists getting together and deciding what's going to happen to the world, and all the real people are out there coming in. And we think of ourselves as being the ones who are outside, and it's the people who are making decisions and not really listening to us. And I think the usefulness of this article and this illustration for me is that how do we break that down? And that's a lot of what the Genetic Engineering and Society Center is about, taking the science and taking public values together to think about how we move forward. So this is what I want to talk about today. And what I want to focus on is a framework that was developed, and Jennifer Kuzma is here today, who's very involved in this kind of thing, in terms of developing a framework for responsible innovation. How do social scientists think about how do you move so that you could improve the way that your science actually gets used? And there are 4 pieces to this framework: anticipation, inclusion, reflexivity, and responsiveness. And I'm going to go through these now, and it's going to be a conceptual thing, but I'll bring them back with the case studies. Okay, and these are quotes from this paper, which is took me a long time to read. It's a different language in a way, but anticipation is recognition of the complexities and the uncertainties of science and society's coevolution. And just think back to the COVID example, maybe. Inclusion, including new voices in discussions of the ends as well as the means of the innovation. Okay, and I'm going to go through all of these just now, but come back to them. Reflexivity, and maybe this is one of the harder ones. Ask scientists in public to blur the boundary between your roles and responsibilities as a scientist working in academia or in a company to the wider moral responsibilities. And then finally, responsiveness. When you have all of this, how do you respond to the new knowledge and the emerging perspectives and views that you bring in? Do you respond? And this could become a cycle of doing that and then more anticipation. So I'm going to focus here on case studies, and they'll be mostly about GMOs, genetically engineered organisms, and this trope that GMOs will feed the world. We know we're having an increasing population size, we have global climate change, and these 4 case studies. So I'll start out with drought-tolerant corn, right? You know that climate is changing. In North Carolina, we just had a drought. Our corn production this year is maybe in many farms going to be only 60% of what it could be. So when I ask people about drought-tolerant crops, most people think that's a good idea. How many of you think that it would be a good idea to have drought-tolerant crops, to breed drought-tolerant crops? How many of you think it wouldn't be a good idea? Nobody. Okay. So I want to tell you a story about drought-tolerant crops. You know, we think of it, and just to say that 2 companies in around 2012 came out with real innovation in drought-tolerant maize, corn. One of them through genetic engineering, one more through conventional breeding, and those were released to the farmers. And interestingly, a paper was published in Science called Greater Sensitivity to Drought Accompanies Maize Yield Increase in the U.S. Midwest. And the title's a little bit funny, but I'll get down to it. But Ron Rejesus in our ag economics department was part of this study, and they came up with a non-intuitive result. And this is the kind of thing that I'll harken back to. They found that there was improved drought tolerance of plants. So if you took the ones that were bred for drought tolerance in a pot and the ones that weren't, and you stopped watering them, it's pretty clear that the drought tolerant ones would be turgid longer, right? And what they also found was when they looked at data from the Midwest, they found that this had resulted in higher average yields for an average year, but there was greater drought sensitivity at the field scale. So what we had done was we took drought tolerant plants and put them in the field in the way the farmer saw, and it led to an increased sensitivity to drought. And this is the deal, right? If you go in North Carolina and you look in a cornfield, the farmers space their seeds certain distance apart, because if you crowd the seeds too close together, the plants will transpire so much water that it induces drought in the field even in a normal year. So you have to figure, there's a lot of things for farmers to figure out. Any of you here who know farming and corn growing, right? It all depends on your specific farm and such. But if you're given drought tolerant seeds, it turns out that you can plant them closer together in a normal year and increase yields, right? So this is what led to the idea that here you had these plants that were drought tolerant, but if you plant them a certain way, there's going to be problems. So why did the farmers do that? And they had a definite logic in doing it. So I want to come back to this issue of responsible innovation. And each of these, I'll just focus on a couple of things. Anticipation, the recognition of the complexities and uncertainties in science and society's coevolution, right? If you're in a mindset that the climate is changing, we need drought tolerant crops, you're going to breed those seeds. But you have to ask what's going to happen when you put it out there. What matters to the farmer? Those few years when there's going to be a drought. And by the way, we have subsidized crop loss insurance, so they don't have to worry as much about that or worrying about the other years. So this is where understanding that interaction, if you're in your lab and you develop a plant that's tolerant of drought, you're so excited, right? And I guess your company is excited and everybody's excited, but you have to think about what is going to happen. And again, including new voices in the discussions, right, about the ends as well as the means of the innovation. What's going to happen to your innovation when it gets out there, right? And so what would have happened if there had been more conversation? I'm not sure if anything would have changed, but at least you would have anticipated what could have happened. All right, I'm going to move to the second case study. And this is engineered insecticidal corn in the U.S. It's something that's dear to my heart because I've been working on it since the late 1980s. And you can see here that the percentage of corn acreage planted in insecticidal corn has gone up dramatically over the years to about 80%. Part of the remainder is because the EPA and USDA require a refuge to be planted in non-engineered corn. But this shows a picture from one of my field plots early on. This is the non-engineered corn, and this is the engineered corn. You can see the difference between them pretty clearly. So this seemed like a really good idea, and it controlled other things than the corn earworm. It controlled the fall armyworm. Okay, the fall armyworm is a pest, has been a pest in the Western Hemisphere, North, Central, and South America, but it wasn't a pest anywhere else. It was confined. But in 2016, it found its way to Nigeria, and from there it spread throughout sub-Saharan Africa. And you can see that these outbreaks and all of a sudden all over Africa. And I took this specific picture of a slide that was in, the Economist, okay? And this is how they describe that. An army of worms is invading Africa. The spread of Spigoptera frugiperta is threatening the region's food safety. I love that The Economist uses Latin binomials. But the final comment in this article was lifting existing bans on GM crops on the continent would lead to fewer hungry caterpillars and fewer hungry people. Here is the solution, right? And the problem is that African countries have not been interested in adopting GM crops, right? So this is the push to adopt them. Very similarly, the Cornell Alliance for Science writes, fall armyworm threatens Nigerian crops. Genetic engineering offers the only long-term solution. So where is this kind of perspective coming about having your innovation and putting it out there? So let me just tell you that there are some issues here in terms of involving other people. This is the approximate proportion of sub-Saharan African farmers growing hybrid maize, 5%, right? But this is what growing maize looks like in Africa. And I think you could talk to the elephants and everything else that goes on there. It's not industrial agriculture. What worked in the United States in hybrid corn, how do you get these into open pollinated varieties? We don't even know how many of these are land races and adapted to specific things. And in order to get stable control of any of these pest populations, you have to put in at least two very different genes. If they're not on the same chromosome, you tell me how are you going to get those into the land races, right? So it's again one of these kind of things where you have a nail and well, you have a hammer and everything's a nail a little bit. So again, going back here, I want to just say that a graduate from NC State, Alan Haruska, got his PhD in 1995 working on the fall armyworm in Nicaragua. And he worked for his whole career in international agriculture. And when the fall armyworm epidemic, or whatever you want to call it, came out, he was charged by FAO in Rome to be in charge of the technical FAO response to the fall armyworm. And they took a completely different approach. Knowing connections with farmers, they took an approach that you would use resources that were already in Africa, and also to look at the excitement about this pest that just got to Africa without its natural enemies, and then watched how the decrease in the damage over time. So Alan was a strong proponent of other approaches here. He then subsequently left FAO and took a job at Michigan State University, but his interactions with all the diametrically opposed groups led him to something very interesting. He published an article in Issues in Science and Technology, which is a policy magazine from the National Academy and Arizona State University. And he took this whole issue of the fall armyworm in this title, What the Global Battle Against Fall Armyworm Reveals About How the US and China See the Future of Global Food Production. And this is a big thing that's been going on in terms of we know that we're going to have a larger population in Africa. And how are we going to deal with improving agriculture in Africa? Is it going to be to support small farms or to move to an industrialized situation? I'm not going to go into this in detail as much as tell you that, there's a real big thing of who's involved in making these decisions about what we're going to do in the future with agriculture. And I just will point here to the same kind of thing. Anticipation, thinking about a product that you have in industrialized country and thinking, all of a sudden plop it down in Africa, where is your anticipation of what's going on? And then again, where is your responsiveness? Once you see what's going on, do you keep pushing this approach to the technology or do you change your message? And that's something that we'll see over the next few years, I think. The next one, third example of a case study. This is a paper that came out in Nature. You may recognize some of these authors. Jim Holland, Ramsey Lewis are at NC State. Ed Buckler used to be here before he made a terrible mistake and went up to Cornell. This paper called Scale-Up Trials to Validate Modified Crop Benefits. Very modest title, but what they're saying is something very important. They, and especially I think it was a graduate student or postdoc who went through all the papers that have been written since the 1990s about moving a gene from somewhere into crop plants to increase yield, so transgenic approaches. And you can see how this has soared over time with over 700 papers published in 2022. And each of these shows an increased yield from 10 to 68%. We're going to be floating in food very quickly. But what their point was that these trials were all done in greenhouses and in small unrepresentative field plots. And I think they should have changed their title. We need to scale up trials to validate modified crop benefits. What they said, and this is pretty obvious, with changing climate and growing population, the world increasingly needs more productive, resilient crops. But improving them requires a knowledge of what actually works in the field. And, that's the emphasis here. And to prove their point, they went through what they thought was a really well-constructed experiment done by Corteva Agriscience that owns Pioneer Seeds. This was a 2-decade project. They tested 1,600 or more genes, and out of those, only 1% of those genes warranted more investigation. And out of those 1%, they subsequently found one gene that could potentially give them what they wanted, the ZMM28 gene. And they increased expression of this gene, and they had a paper finally published in PNAS. They had tested 48 hybrid plant types, 4 years in 58 global locations, and their finding was that ZMM28 could increase yields by about 2%. Remember, 10 to 68%? This is a big finding. 2% yield increase is nothing to, that's good. Though conventional breeders tend to get 1 to 2% increase in yield per year. So again, I want to bring this back to these issues about responsible innovation. And this is a place where reflexivity is really important. Think about yourself as an academic scientist or in a company, and you're trying to get tenure or whatever it is you're doing, and you want to get your paper in a high-profile journal. When do you publish? Do you wait until you've had actual field trials for 3 years, or do you publish after your first greenhouse experiment, the one that looked good, right? And then responsiveness is the same thing, right? How do you respond to this paper, right? In this paper, it's very modest in some ways, but it really says to the reviewers and the publishers, right? Change your model, right? If you get papers like this, don't just have them reviewed by molecular biologists, have them be reviewed by the people who are actually working on the ground. So I'm going to move to the fourth one, and I'm not going to go into too much detail, except that we've done a huge amount here at NC State. Many of you have probably heard about gene drives for agriculture, environment, and human health. The idea that you can use genetically developed super Mendelian inheritance so that you could develop a genetically engineered pest, whether it's an insect or a rat or whatever, release it into a population, and it will, because of super Mendelian inheritance, push itself into the population, even if it decreases the fitness of the individuals. And some of you here know very much about that. And I have to say that the approach uses CRISPR-Cas without getting into too much detail. Instead of just putting a gene into one chromosome, once you put that in, that gene itself moves it through a cut site and makes heterozygotes into homozygotes, thus doubling the number of copies. So briefly, if you have normal inheritance and you put a gene in a heterozygote condition, only half the offspring will get it. But if you have a gene drive that makes that heterozygote into a homozygote, all the individuals will get it, and slowly it will penetrate the population, even if it's suppressing the individual. So that's one of the goals, especially with crop pests and so on. So we've been involved with this for a long time. Jeff mentioned that we had a grant from the National Science Foundation training graduate students, and this was one of our things about getting into this kind of responsible innovation and bringing together faculty and graduate students from the social sciences and policy and ethics, as well as population genetics, ecology, and molecular biology to sort of all work together in this project. And that was very successful. The graduate students had to interact with each other in hard ways, right? If you're in social science and genetics and all of those things and work together on projects, and they've gone on to their interesting, diverse careers. And we got a subsequent grant to go back to the crop issues. And that's actually going to end in a couple of weeks, but we fortunately have some university investment in this for the next couple of years. But that whole thing has still got a group of faculty and postdocs and all who are still got active projects in this area, even though we haven't had a single release of a gene drive organism yet. Looking at this from so many perspectives is what I would call responsible innovation. So I'm not going to go into this in too much detail because I want to get to the next piece of this talk without keeping you past 5:30. And that is dealing with genetic determinism. And this is really important. This is where the past comes in. And how is NC State facing this issue? And this is where we have to go back to our academic ancestors. Some of you know all of the people on this slide, but just quickly, Galton is sometimes considered the father of biometrical genetics. Pearson, how many of you have ever done correlation analysis? Yeah, most of you use Pearson product moment correlation. That's the guy. Punnett. I know you all went through high school, so you know all know what a Punnett square is. And then how many of you have ever done analysis of variance? ANOVA? Yeah, there's the man, R.A. Fisher. That's, yes. And then there's Herman Muller. He won a Nobel Prize for using X-rays to create mutations and map them. But I want to tell you more about these ancestors. Galton was a cousin of Darwin's, and 20 years after Darwin's book came out, he wrote a book called Hereditary Genius, which set out the rules for eugenics. Pearson was the initial editor of the Annals of Eugenics. These are from our library. This is the first journal publication. And you can see Annals of Eugenics, a journal for the scientific study of racial problems. And he used all of Pearson product moment correlations to study these issues. This is the first paper in that, authored by him and a colleague, The Problem of Alien Immigration into the Great Britain, illustrated by examination of Russian and Polish Jewish children. Right? Punnett wrote a paper called Eliminating Feeble-Mindedness, in which he criticized the eugenic approaches for getting rid of feeble-mindedness as taking 8,000 years to accomplish. He wasn't against eugenics, he just thought it had to be done better. But Fisher didn't leave it that way. He wrote a paper where he insulted Punnett as having tables in which Professor Punnett has inadvertently supplied material for anti-eugenics propaganda, right? So much for subjectivity in science. And then there's Herman Muller, who hated capitalism. He left the US and went to the Soviet Union, worked with Vavilov and others, and wrote a book, Out of the Night, which pushes for positive eugenics. And he actually wrote a very long letter to Stalin, and just taking out a little pieces of this. Oops, I thought I had something underlined. This is a different presentation that I thought I was going to be giving because it's a different version. But just to say, scientific application of genetic knowledge unhampered by bourgeois social and ideological fetters, on the other hand, it will be possible with only a few generations to bestow the gift of so-called genius upon practically every individual in the population. This is what he's writing to Stalin. And what he's suggesting is that proletariat women should be breeding with scientists. Yeah, pretty interesting. Stalin didn't like that, and he had to leave the Soviet Union. But I mean, just to give you a tinge of where we're coming from, right? And agriculture is always brought into this, right? If we can breed plants, we can breed humans, right? Only healthy seed must be sown. Check the seeds of hereditary disease and unfitness by eugenics. And even after World War II, after the Nazi Holocaust, this is Life magazine. I don't know how you would say it in today's language, but everybody got Life magazine. Eugenics in a cornfield, still with us, right? Bred right, grown right. This is some of the detail of it. So after the 50s, we thought we were moving away from this. The annals of eugenics changed to the annals of human genetics, and maybe we're moving on. We had new luminaries, Stephen Jay Gould and Richard Lewontin. Stephen Jay Gould wrote a book in which he debunked about 300 years of the mismeasurement of man, the whole idea that there were racial differences and there were superior and inferior races. Um, and then Richard Lewontin, who took in 1972 all the data available on human population genetic variation and looked at where it came from. And in this spread of genetic variation, 85% of the variation occurred among individuals within populations. An additional 8.3% came from different populations of the same supposed racial combination. And then 6.3% of the variation was among population of different races. And again, races, I say that word, but you can't from this picture decide that you actually have races. But that died down, and based on some conversations with Henry, I felt obligated to go through a book called The Bell Curve: Intelligence and Class Structure in American Life. Like Stephen Jay Gould and Lewontin, Herrnstein was an endowed professor at Harvard. Charles Murray graduated from Harvard, but worked for the Heritage Foundation, right? This 845-page book, that's why I didn't read it right away, right? Includes 292 appendix pages and 94 figures and tables. A very involved book showing that intelligence and class structure in American life will remain consistent. And these are some of the quotes I took out of this book. The difference in IQ scores among racial and ethnic groups are largely attributable to genetic factors. Okay, and they had lots of studies in this book, and this is showing the distribution of IQ in black and white populations in the United States. This book is a very long book, and they had plenty of chance to contradict themselves. So another interesting thing was in another quote says, individuals differ in intelligence due to a combination of genetic and environmental factors. I even saw a part of the book that said, well, it may all be environmental factors that are causing the differences. But then they say that the impulse to think that the environmental sources of differences are less threatening than genetic ones is natural but illusory. This is very important. Number one, they call it threatening. And number two, they call it illusory, because what they're saying is that even if the differences was caused by environmental differences, we won't be able to change that. That's basically what they're saying. And then in their conclusion chapters, this is the very end of the book, it says, a society with a higher mean IQ is also likely to be a society with fewer social ills. And the next sentence is that the most efficient way to raise the IQ of society is for smarter women to have higher birth rates than duller women. Pretty strong statement, okay? This is with us, right? And this was picked up. You know, people were waiting for this book. Not everybody, but people were waiting for this book. And it went out in social media. And this is baked in 1994. Things, you know, we've made a lot, you know, there's like this pendulum swing. But curiously, in 2015, Gregory Clark published this book, The Sun Also Rises, a takeoff on Hemingway's The Sun, S-U-N, Also Rises. Again, plenty of data indicating that social status persists within families over many generations, far longer than conventional estimates suggest. and that environmental factors, family size, and racism plays less significant roles in social mobility than previously thought. That was the premise of this book. And that book also got a lot of media attention, and Gregory Koch likes to get that kind of attention. But the reason I'm bringing this up as much today as I am is because in 2023, Gregory Clark published a paper in the Proceedings of the National Academy of Sciences. This is a journal that I do reviews for and edit, and I don't know how it got here, but basically saying that class structure in England in 1600 and 2022 is about static. That with all the social programs that we've tried to institute since that time, that there hasn't been a change. And inheritance, he admits that it's correlation, not causation. But if you read the paper, I did a word count, genetic and genetics 56 times, non-genetic once, and environment 3 times in terms of mentions. And what's really disturbed me is that 1 out of 20 papers in Proceedings of the National Academy gets a commentary because of how important it is. And this paper got a commentary. And the commentary was stronger than the paper itself. The heritability and persistence of social class in England, arguing even further that the reason that you have these social classes is genetic. There are some people who just have the right genes. And I'll just show you, I told you there'd be one thing. They used Fisher's equations for not just heritability, but also putting in the genetic correlation between mates. And that leads to greater persistence. And they fitted their data, and they had lots. This is a big study, and I don't want to go into a huge amount of detail, but if you look at it, this is their correlations. They had this looking at correlation occupation status against implied shared genotypes. So, right, if you're looking at a child or siblings, they're going to have a lot of genes in common, is the way they're saying it, and their occupations are going to be correlated. So if you have 2 children, right, they're going to be high or low, they're going to be correlated. And as you move to cousins, 4th cousins, they're not as related to each other, and there's less of a correlation in their occupational status. When I read it, it was pretty clear to me that all of this could probably just be due to environment. And I looked for papers that rebutted it, and I only found one in bioarchives. And many of you may know Ruth Shaw. These are really good quantitative geneticists, and they took this paper apart. Confounding fuels, hereditary, and fallacies. And they showed that this could all be environment. Unfortunately, they haven't been able to get this paper published. There's a lot of politics in science. They were able to show that if you just forget about the shared genotypes, if you just looked at correlation in paternal wealth, right? If you're a full sibling, you only have one father, so the correlation is 1, right? And if your grandparent, parent, child, that's going down, the correlation between the parental wealths, right? And if you go to 4th cousins, the parents may not have the same wealth at all, and therefore the correlation in occupations. So I have a different interpretation of the title of the original paper, The Inheritance of Social Status. Yes, it is the inheritance of social status, the inheritance of money from your dad that determines your social status. Right? So not only this, but they also showed that the paper showed that there was no change in social mobility. When they took out pseudo-replication out of the study, as you can see with each of these things that they studied, there was a change. So I just want to come to this and talk about how this connects to the Genetics and Genomics Academy, in the sense that these things are not going away, right? These things are going to keep bubbling up, and some people are going to want one science and some people are going to want another science. I'm not saying that genes don't determine your intelligence or something like that at all, but the issue is, what is it going to matter which way it goes? What are we going to do different as a society depending on whether it's 10% or 20% or 80% genetics versus environment? So, but I guess I just want to get to what are we doing here at NC State? And I just want to give a few examples. During the COVID epidemic, we had Angela Saini come and talked online on the legacy of scientific racism, a book that she had written. We had over 1,000 people connecting online, and the provost gave introduction to that. We had Latifah Jackson here talking about the implications of environmental stress on human health as opposed to the ideas of what's going on with genetics in human health and racial differences in human health. And recently we had Brian Donovan come here, and we have an ongoing relationship with him in terms of how to teach genetics at the high school and middle school levels to decrease the ideas about white supremacy. And he just had 2 papers published in Science on these studies that are really detailed research studies. So these are the ways that we're moving, but I do want to give some shout outs to things that are happening. This year, as you heard, we're going to have we have a 3-credit course that's already started with full enrollment, 75 students that Claire and Melissa and Dahlia are teaching, and it was taught actually last year by Joe Graves, who wrote one of the books in this area. I also want to give a shout out to the Genetics and Genomics Education Research Interest Group, which, I mean, if you want to think about how you're going to get past this kind of thing, equity in terms of education and bringing in people from all groups to feel welcome is so important. And finally, to say also, you know, we did submit this proposal, and the title of our proposal is Genetics, Genomics in Societal Context. So again, trying to bring responsible innovation as we move forward. Hopefully we'll get funded. We'll have 44 of our GGA trainers involved, but we're also going to have the genetic engineering and society, social scientists and humanities people working with us on that. They are just the ones who are really going to make education here a difference. And just to bring up, this has been talked about before, and other piece that we have to think about is somewhat separate. It's about evolutionary biology, which is probably the most controversial subject that you could have, right? And I don't think we've really figured out how do we teach our religious students about that in a respectful way? How do we move that forward? And Ken Miller came here in 2006 and gave a lecture on this. We had a lot of interaction, and we're really proud that we're going to have him come back in October. I hope you'll all work on that. So again, I want to just end by saying that this is a big challenge for us. And today was really great for me to see all of the intellectual rigor and the humanity that comes out of this group. And I think that we have challenges, and moving forward, especially by having more collaborations with the Genetic Engineering Society Center, I think we can accomplish a lot. And finally, just a shout out to Jeff Yoder for taking over leadership of the GGA. And I can see already moving in this direction and bolstering and moving forward. So thank you very much. Right? Time for beer or whatever, right?
So thank you again, Fred. I really appreciate not only the seminar here, but all the efforts that you put into building and the leadership that you put into the GGI and the GGA, and your shout out to the executive committee. I'm 100% with you that I would not be wanting to step into this position if it weren't for the executive committee already being here, that, you know, knowing the potential for moving things forward and building on what is already here. So thank you. And then I do want to announce that this has been the first annual Fred Gould Genetics and Genomics Keynote Lecture. This lecture series if we haven't embarrassed you enough, hopefully we have now. This lecture, we're doing this named lecture series moving forward after Fred, and it's going to be integrated into all future GGA retreats where we will invite speakers, and that will be the name of that series. And we want to thank you, of course, for all of your support and your career-long commitment to not only genetics and genomics, but also to the university here as well.